Alright, so if you're wondering where I'm getting these questions from, Cambridge O level. Alright, so we have this interesting question which you need to know, and these questions are highly examinable, coming from your specialization, and you need to know them. So figure 1 shows an a red blood cell and a elute air cell. So we have a red blood cell, a elute air cell. Now there are some distinct features. That's the reason why I'm saying this is an interesting question. You and I will know to say that a red blood cell doesn't have a nucleus, and this is because the absence of the nucleus creates the space for carrying hemoglobin, which have the high affinity for oxygen. So this is where now hemoglobin is carried. So that's the reason why we usually say the red blood cells, they are biconcave in shape. They are like this, even this side like this. So they are biconcave in shape, or biconcave disc. That is biconcave, so they lack the nucleus. But as you can see here, this elute air cell, it has a nucleus. So those are the differences that you need to be aware of. Then in terms of the vacuum, the red blood cell doesn't have the vacuum, doesn't have the cell wall. So let me start with this one. This one is called the cell wall, so that we just make things easier. Cell wall, cell membrane. Some they say selectively semi-permeable membrane is just okay. And this one is cytoplasm. So cytoplasm. And this one is called the vacuum. Now one thing that you need to observe here is that, and this is the nucleus, that one thing that you need to observe here is that this one is a elute air cell. So elutes are just found in the ground, isn't it? So they are not exposed to sunlight. So one thing that you need to see here is that these elute air cells, they lack chloroplast. They don't have chloroplast or chlorophyll. Sometimes you can say chloroplast or chlorophyll, whichever way you like. Chloroplast, chlorophyll. Yeah. So that is how it is. Let's write it nicely. Chloroplast or chlorophyll. Alright, so let's see the questions. So I have the first question, which is saying select 2 structures in the elute air cell which are also present in the red blood cell. In each case, state the letter, that is A to E, and name the structure. So if you look carefully here, what structures are present in both a red blood cell and a elute air cell? So let me start with the cell wall. Doesn't present. The cell membrane is present. The cytoplasm is present. So we have B and C. So here we can say B. And what is structure B? The cell membrane. Then the second one is structure C. What is structure C? The cytoplasm. Then the next one is saying name one structure which is typical of many plant cells but which is not present in the elute air cell. So as you can see here, like I said, chloroplast or chlorophyll is not present in the elute air cell. These are not exposed to sunlight, so photosynthesis can't occur here. But you're going to find like there are some elute air cells, not like those which are found under, those which appear at the surface. They look green because they are exposed to sunlight. That's the main reason. So here we can just say chloroplast, or you can say chlorophyll. It is also okay. Then let's try to answer this one, an interesting question under magnification. A student measured the length of a structure using a microscope. So the magnification used was 250 times. So this times is just a unit for magnification. You can either put it here or at the end of a number. So the image size of the structure was 5.00 millimeters. What was the actual length of the structure? So as you can see here, we have millimeters, meters, or micrometer, micrometer, and micrometer. So the one which is dominating here is micrometer. Alright, so the formula is magnification being equal to, but when it comes to structural questions, you're supposed to write it in full, not in abbreviation. So magnification is equal to the image size. The image is the one that you draw or you see under a microscope. The image size divided by the actual length. So as we are finding the actual length, so I can make actual length the subject of the formula. Okay, so let me just do this. So what do we have? We have the image size, which is 5 millimeters, and then the magnification here, it is 250. So I can say here I'm going to have 1, then here I'm going to have 5, and it is going to be 15. When I divide, I'm going to get 0.02. Now this is in millimeters, but if you check here, we don't have any answer which is in millimeters as 0.02. So what are we going to do? 1 centimeter is equivalent to 10 millimeters. Then 1 meter is equivalent to 100 centimeters. So if you want to convert this one here into meters, we need to divide it by 1000. So 0.02 divided by 1000. Then what am I going to get? I'm going to get 0.00001 and then 2 millimeters. Still we don't have this one here, but we can convert this one here into micrometer. What is micrometer? Micrometer is more like you are having something like 10 to the power negative 6. That is micrometer. Okay, but here what do we have? How many decimal places do we have? So if I start counting from here, 1, 2, 3, 4, 5. So we have 5. Okay, so we have 5. This one is not accommodating to 6, isn't it? So what we are going to do for us to make sure that this one here is converted into micrometer, we are going to write 2.0 multiplied by 10 to the power positive 1, then micrometer. So as a result, if you try on the calculator, from saying that micrometer is more like 10 to the power negative 6. So if you multiply 10 to the power negative 6 multiplied by this one here, this one is more like 20 micrometer, because this one is positive. You're going to find that you're going to get the same answer. So this is our final answer. That is how you're supposed to answer. Thanks for watching.